Have you ever looked out at a field, just a huge meadow, and tried to imagine counting every single plant in it? Every blade of grass, every wildflower? I mean, it sounds completely impossible, right? But for scientists, getting that count right is absolutely critical. And it's led them to some surprisingly simple and, frankly, elegant solutions.
So let's put ourselves in their shoes. If you're an ecologist and you're tracking the health of an ecosystem, how do you get a truly precise measurement of what's growing there? I mean, a single square meter of a meadow can be this beautiful, tangled, chaotic mess of dozens of different plant species, all overlapping. How in the world do you turn that into hard data?
Well, the one thing you can't do is just eyeball it. You can't just glance over it and go, yeah, that looks like maybe 30% clover. Our eyes are just way too easy to fool. In fact, a study back in 1991 looked at this exact problem and found that visual estimates have error rates that are just way too high for any serious science. For a single person, just repeating their own estimate, the error could be plus or minus 18%. And between two different people, it could be as high as 24%. If you're trying to track small changes from year to year, that kind of error margin just makes your data useless.
So yeah, that brings us right to the heart of the challenge, what we're calling the meadow measurement problem.
And just look at this. This shows you how seriously scientists took this problem. This is not something you just buy at a hardware store. This is a custom-built rig from that same 1991 study. You've got tripods, a linking bar, a water level. I mean, the whole thing was designed with one and only one goal in mind: to hold a single steel needle perfectly steady and lower it in the exact same spot over and over and over again. It's really a testament to just how much precision matters.
So you see a complex-looking machine like that, and you'd think the solution must be incredibly complicated. But it's not. Actually, the method that whole apparatus was built to serve is, well, it's beautiful in its simplicity.
It's called the point-intercept method, and the idea is just brilliant. You lay out a line, or maybe a grid, over the area you want to measure. Then, at very specific regular intervals, you lower a very thin pin straight down. Every single thing that pin touches on its way to the ground, whether it's a leaf, a stem, a flower, is recorded as a hit for whatever species it belongs to.
And that's basically it. You lay down your measuring tape, that's called a transect line. You lower the pin at, say, the 1 foot mark, you record the hit, then you move to the 2 foot mark, lower the pin, record the hit. You do this hundreds of times, and by the end of it, the number of hits for each plant divided by the total number of points you sampled gives you an incredibly accurate percentage of its cover in that area.
And this right here is what makes it so powerful. When you're doing a visual estimate, a scientist has to make a judgment call. But with the point-intercept method, the only decision is, did the tip of this pin touch this plant, yes or no? It totally removes that subjective guesswork from the equation. As the U.S. Forest Service Guide puts it, it's one of the most trustworthy and most nearly objective methods there is.
So you don't have to take my word for it. This whole method is backed by some really rigorous data. This graph here comes from that same 1991 paper. What the researchers did was they tested the method's accuracy by sampling the same exact plot 10 different times. And what they found was this really clear relationship between how often a plant appears and the expected error in measuring it. This is huge because it lets scientists actually quantify their own accuracy, proving that sampling at these fixed points is way more reliable than just random sampling or, of course, visual guesses.
All right, so we get the theory and we've seen the proof, but what do scientists actually use when they're kneeling in the dirt? Let's take a look at the toolkit. First up is this guy, a point frame. Instead of lowering just one single pin over and over again, this frame lets you lower 10 pins all at once. You can see how this would be a huge time saver. You just place the frame, lower all the pins, record all the hits, and then move the whole thing to the next spot. It's a really efficient, practical way to use that core principle. And here's another clever variation, the grid frame. See here, there are no physical pins being lowered at all. Instead, the points are just the intersections of the strings or the wires. A researcher looks straight down through the grid and just records whatever is directly underneath each of those crosshairs. This is great for taller plants where a physical pin might get a little clumsy. It just shows you how adaptable this basic concept really is. Now, this is one of those little details that makes a huge difference. When you're working on a slope, you don't lower the pin perpendicular to the ground. You have to lower it perfectly vertically, straight down towards the center of the earth. So why does that matter? Well, because what you're actually measuring is the plant's canopy as if it were projected onto a perfectly flat map. If you lowered it at an angle, the pin would travel through more vegetation and it would artificially inflate your measurement. It's exactly this kind of strict, consistent method that makes sure the data is accurate and comparable anywhere in the world.
Okay, so for decades, this was pretty much the gold standard. But, as technology evolved, so did the method. And this is where we take the leap from the physical world into the digital one.
This slide just illustrates that digital leap perfectly. On the left, you've got a physical transect line out in a real field. On the right, you've got a digital photo overlaid with sample points. The principle is 100% identical. Instead of walking along a tape measure and physically lowering a pin, you are now looking at a picture of that same ground and dropping digital pins, pixels, right onto the image.
And the tool that makes this all possible is this really brilliant piece of free software called SamplePoint. It was developed way back in 2004 by some researchers with the USDA, and it was specifically designed to put this powerful scientific tool into the hands of absolutely anyone for free.
And the way it works is incredibly simple. You just load a high-resolution photo of your meadow, or, a coral reef, or a patch of forest floor. The software lays a grid of crosshairs right on top of the image. You then just go point by point, identify what species is under the crosshairs, and click to classify it. The software does all the hard work, automatically tallying the results and saving them right to a spreadsheet. It is the point intercept method, perfectly translated for the digital age.
And this is not just some novelty toy. If you take a quick look at the SamplePoint website, you'll see a list of over 50 peer-reviewed scientific papers that have used this exact software for their data collection. It's been used to study, well, everything, from sage-grouse habitats and pipeline reclamation sites to Arctic vegetation and weed diversity. It is a proven, accepted tool in modern science.
So, this brings us to the big final question. In a world of satellites and drones and AI, why does this simple, almost old-fashioned idea endure?
I think this quote from the USDA really says it all. In science, objectivity is everything. You want to remove human bias and human judgment as much as you possibly can. And this method does exactly that. It transforms a complex visual scene into a simple, binary set of data. Was it a hit, or was it not a hit?
So when you boil it all down, its success really rests on these 4 pillars. It's objective, which we just talked about. It's highly accurate, which has been proven by decades of research. Its core principle